All right, it's 2 minutes past after. Um, all right, let me welcome everybody again, and welcome to the 2021 GemPharmatech webinar series. Today's webinar is special, not only because it's the first one of the year, it also marks an important milestone in GemPharmatech's global footprint, the establishment of a fully owned subsidiary in the United States, GemPharmatech US. For this special occasion, CEO of GemPharmatech US, Dr. Mark Moore, will join us to share an introductory presentation of the company, as well as his vision leading GPT going forward. For the interest of time, I will introduce both speakers of the day at the same time. Dr. Moore completed his undergraduate study as a biochemistry major at Princeton University and received his PhD in molecular biology and immunology from Brandeis University. Shortly afterwards, Dr. Moore embarked on his journey of entrepreneurship, first as a senior scientist at Genentech, then as CEO and co-founder at Daltogen. Following these milestones, Dr. Moore played a major role in the NIH initiative, the Knockout Mouse Project, KOMP, and served as an executive director for the International Mouse Phenotyping Consortium, IMPC. For anyone in the field of genetically engineered mouse models, whether as a user or a model provider from academia or industry, both KOMP and IMPC are familiar names. Before joining GemPharmatech, Dr. Moore was CEO at LifeEdit and Gliologics. Our second speaker of the day is Dr. Pradeep Kumar Sasasaran, Vice President of Business Development. Dr. Sasasaran has a PhD in molecular and cellular medicine from the University of Oxford. He was a Fulbright Scholar at Harvard University, a Daniel Turnberg Fellow at the Hebrew University of Jerusalem, and the Ambo Fellow at the Sorbonne University. Before joining GemPharmatech, Dr. Sasasaran has served as an honorary lecturer at the University of Liverpool and a faculty position at the Xian Liverpool University. Today, Dr. Sasasaran will discuss the Knockout All project at GemPharmatech. Without further ado, please join me and welcome Mark.
Thank you, Jing. It's an honor and a privilege to be here today. I'm very excited to be joining GemPharmatech and help bring forward the amazing research tools that have been created at GemPharmatech in China to the US research community. What I'd like to do today is go through a little bit of the background and the history of both GemPharmatech and some of the brief history of genetically engineered mouse models. So as many of you may know, that virtually 90% of all Nobel Prizes that have been awarded in physiology and medicine have involved animal models. And that is one of the most exciting underpinnings and the need for better research models to enable research discoveries as well as better pharmaceutical development. This really hit the forefront in the late 80s with the development of embryonic stem cell technology and the ability to really genetically engineer mice beyond random transgenics, which started really in the late 70s. With this advent of ESL technology, around 2005, Francis Collins, who was then the director of the Human Genome Research Institute, and had recently published the first draft of both the human and mouse genome, assembled a team in Cold Spring Harbor to discuss what could be done with better mouse models, because many were being made in the research community. They were not standardized, and in fact, they were difficult to acquire. Many people would spend a year or two developing mouse models with ESLs, and then basically would continue to work with them and not necessarily make them available to the general public. So with this meeting called the Banbury Conference, it was decided that it would be worthwhile for the US, with assistance from other interested parties, primarily in Europe at this time, to develop an entire library of embryonic stem cells where each gene that encodes a protein in the mouse genome would be knocked out, and this would then be made available to the entire research community, both public and private. The goal then is that these would then be developed into mice. There would be some phenotyping analysis, and then this data also would be provided to the entire public. However, at this time, the funding really only covered making a library of the embryonic stem cells, and I was quite happy to be involved in the Knockout Mouse project, both at its inception and also managing the first 5 years of that program in combination with my colleagues at NIH, particularly Colin Fletcher. 5 years after the initiation of that, we were able to get additional funding of over $120 million from the US NIH group to develop the International Mouse Phenotyping Consortium, which really was kicked off by the MRC Harwell Group and the Sanger Institute as well as the foundational members of this phenotyping consortium. I was very honored to be selected as the first executive director of the IMPC. And with that, we recruited a number of different groups, which eventually totaled 16 different research institutes around the world in 13 different countries. And so this network would then take the embryonic stem cells and create mice and provide a phenotyping analysis of them, which is in place into a database and then presented to the entire scientific community. You were also able to order the live mice from the various members of the IMPC who had created those mice, again, enabling research throughout the world. This is very exciting, and I think it was going quite well. And then in 2012, the world changed, and the world changed with the CRISPR craze. We went from having this entire collection of embryonic stem cells, which were primarily knockouts, which had a reporter gene in them, to the ability to create knockouts or other mutations in actual zygotes in the mouse embryo. This saved nearly 6 months of time in creating the mouse models, and also despite the great utility of embryonic stem cells, virtually half of them did not go germline, so we could not make a mouse. So the cost was tremendous. The advent of the CRISPR technology really allowed us to cut the cost by a factor of about 2/3. So with that, we moved from the embryonic stem cell, and the IMPC started making mice directly with these knockouts. Unfortunately, at that stage, the technology was still difficult and expensive to do things such as conditional knockouts or inserting reporter genes, so we created a library of knockout mice with the IMPC. In parallel with this, GemPharmatech was a spin-out from the MARC, the Mouse Animal Research Center in Nanjing, which was a member of the IMPC. And the MARC group, which later spun out GemPharmatech, started advancing CRISPR technology to do model customization services, adding conditional knockouts, humanization, creating immunodeficient mice, and developing preclinical efficacy and testing, as well as mouse breeding services. So in essence, what has been able to be accomplished at GemPharmatech is really that initial dream that we had in 2005, which is to make any modification in the mouse genome at will and provide better mouse models for better pharmaceutical development. And what we will do today is focus in on one of these aspects, and that's really the conditional knockout mouse program. But GemPharmatech as an organization offers all of the different services and products that you see on this. And our goal in the US, as I mentioned, is to expand this to continue to create better mouse models for human disease, better mouse models for better pharmaceutical development. And again, we believe that these technologies have really allowed us to create amazing tools to accelerate research and discovery. And I'm very excited, having worked in this field for more than 25 years, to see how quickly and rapidly it has grown. And I'm excited and really believe that in the next 20 years, we're going to see amazing breakthrough therapeutics that's unparalleled because of these better mouse models to create, again, better pharmaceuticals. And so with that, I'll turn that over to my colleague to give you better details around this program. Thank you. Pradeep?
Well, thanks, Mark, and thanks, Jing, for that wonderful introduction. I'm just going to share my screen now with my slides. Hopefully all of you can see it. So as Mark mentioned, one of our flagship platforms is the Knockout All project. Our abbreviation for it is the KOAP. And I'm going to talk about this in detail today and what it's all about. And I'm going to essentially answer three questions. What is the KOAP? Why are we doing it? And how are we doing it? So these are the three questions we're going to touch upon. That's basically a lovely picture of our headquarters in Nanjing, if you're wondering. So first question, what is the KOAP? Well, as I mentioned and Mark mentioned, it stands for the Knockout All Mouse Project. And it's our ambitious target of producing conditional normal knockouts of 22,951 protein coding genes, and also making these tools available readily to the scientific and academic community. And this eventually will become the largest resource bank in the world for this type of mouse models and platform. So that is what, but why? So I know there's some Chinese and some Far Eastern colleagues joining us. And also we were pleased in round one, there were some public members joining as well. And what we have in the West is a saying, be a kid in a sweet shop. That's really the mentality I wanted to portray to you all today, and what allows GemPharmatech to be a world leader in this space. So I, as my family and friend knows, have a big sweet tooth. I love my sweets, and I've been lucky to travel the world trying different things and different sweets in different places for my postdoc travels. And when you are a kid in a sweet shop, your eyes open and you want to grab everything, you want to try everything, and you have freedom. And that's the kind of mentality we want our customers, our consumers to have. So if you're a scientist and you want to try this knockout, you want to have freedom and you want to take risks, that's what GemPharmatech's here for you to have the mentality of being a kid in a sweet shop. And this is what I'm going to touch upon later on. And the second, obviously, why is that it allows our academics to do their work, but do their work at a high quality. So far, I mean, this is still an old figure, everyone. So far, we produced over 170 publications just using our KOAP mice. I should say we, meaning our customers. And these were published in some top quality journals. So why we're also doing it is that it allows academic freedom and publications and a knowledge database that can be shared across the world. So that's the why and that's the what. Now I want to go detail into the how. So I know there were some members of the public joining us, so I'm going to keep this very fundamental at the start. What is the KOAP? How is it achieved? Well, we know there's three different kind of ways to go about genetic modification. You have the spontaneous mutation, the random induced mutation, and also gene modification targeting itself. And this gene modification targeting, as Mark mentioned, is the key. So there's really four different types. You have the knockout, which can be a knockout traditionally or a conditional knockout. For those who don't know, the conditional knockout means that we have spatial and temporal ability to govern how we delete certain genes. So we can delete in a specific tissue when we want and where we want. We also have the knock-in, so we can add genes and do point mutation. We also, a different kind of gene modification is when you have multiple targets. And as Mark mentioned, we can have humanized modification where we can have human genes in mouse to study the development of disease better. So we can do complex modification as well. And then we also have the transgenic overexpression ability to overexpress certain genes to see what happens if these genes increase their activity and what happens if these proteins also increase their activity. So there's several different ways. The Knockout All project obviously concentrates on knockout mice, but just to make this clear, we at GemPharmatech are experts at all these facets and platforms. So we can really work with you on all these different four angles as well. So again, Mark stole most of my thunder, but what we're looking at is the early 80s where the mice were describing knockout models themselves. And then eventually the Nobel Prize winners were announced in 2007. And what we see, the traditional method was making a vector construct and using ES cell culture to develop these vectors and grow them, and then inject them in blastocytes, and then you can have eventually upon breeding and animal husbandry, you can have these mice developed in terms of knockouts. So that was the traditional way. And ever since then, we had a flood of different technologies like the ZFN and Talons, but the CRISPR-Cas9 is certainly the one here to stay. Last time I was seeing a series online in Netflix, and they mentioned CRISPR-Cas9 as it was nothing. So even the public, and it's becoming a norm in everyday life. So the CRISPR-Cas9 is a technology that we actually embrace straight away in GemPharmatech, and we're very proud of it. So we've been developing it rapidly, efficiently, and at a scale that we think is well beating. And essentially, it allows us to save time, do this transfer and rederive quicker. And what does this mean? So it means if you look at the advantages of CRISPR-Cas9, what we use in the KOAP project, again, as Mark mentioned, it saves a lot of time. So traditionally it might be 14 to 16 weeks, the first step here, and then the 24 weeks subsequent. But with CRISPR-Cas9, where we can cut that nearly over half, and the first step is only 2 to 6 weeks. So it reduces the timelines very generously, and also allows us to make models of knockout mice in various different backgrounds. So these are the real advantages and how we have implemented this at GemPharmatech. And again, if we look at the data that comes out in terms of birth rate and the project types, and comparing it to both. So we look at a CRISPR-Cas9 in the first column and the normal germline rate here. You can see the conditional knockout, we have a birth rate of 12% compared to a 2.4%. And this is drastically increased at 20% compared to 7.5% for the conventional knockout. So again, there's another clear advantages in terms of using CRISPR-Cas9 technology. So we've talked about the how. So how now do we implement this, and what are our results so far? So exponentially, we have immense growth in our mouse strains. We're currently totaling over 15,000. We're trying to flux, as I said, and knock out all these genes, protein coding genes. Currently, we sit at 7,792 genes targeted, and this rate itself is growing rapidly. And what's fascinating and even shocked me when I joined the organization is the pure scale of how we can do this rapidly. And now we can make and generate these mice up to 3,000 over 3,000 strains per year. So that's a fantastic achievement from GemPharmatech, and we're just building upon this every year. So I get a lot of customers and scientists saying, "Hey, Pradeep, we like your library, but what's in the library? Am I important? Am I mice? Is my research important? Is my kind of a field important?" Well, all of you are important to us, and we're going to have 3 slides now to break this kind of strategy down. So if you look at gene families and what we're targeting, we target a very varied kind of proteins and genes, and we have no specialist disciplines or targets, unlike some of the other contract research organizations. By far, obviously, our knockouts and conditional knockouts so far are GPCRs targeted, but we have other things from cyclins to ion channels to receptor kinases, and our growth is just phenomenal. And our conditional knockout strains is actually quite large in terms of strain numbers as well. So that's something to note. And then we're not just limited to gene families, and we're not limiting ourselves to certain things. So if you're a cellular biochemist, and if you want to look at certain proteins in the cell itself, we have a varied range of products here and mice strains. So if you look at protein subcellular localization and what kind of strains we have, it's off the scale. I mean, I wish I had this when I was a PhD student, because I was looking for some of these cell-specific knockouts as well. So we can look by far the nucleus targeted proteins. We have up to nearly 5,000 strains. We target proteins in the mitochondria, the plasma membrane, even the microvilli as well. So here again, we have a subsequent kind of varied and library here for your use. And then another question is, pretty, I'm doing aging research, I'm doing auditory research, I'm doing hearing research. Am I important? Yes, you are. You are important to us. And we have a varied amount of mice in different fields. For example, if you look at aging field, which is close to my heart, I did my PhD and my postdoc research in aging field. We have knockout mice up and conditioned knockout mice associated with the aging field, nearly 3,000 different strains in that field itself. And even the auditory field, which might be small in different competitors, we have up to 500 mice targeting different proteins. So again, here you can see that we are developing strains across the spectrum of fields and across the biology as well. So that's something exciting for you guys to toy away with. And we don't just stop there. We're trying to make life as easy as possible for you. So our website is world class. We have some new techniques we're implementing. Just like if you go on Amazon or eBay and you're doing e-commerce, we have this a website that's very user friendly. So you can use a shopping cart kind of mode where you can add things, take things out, and you look at different products and really go by one and see what happens. Now, a little apology. We're trying to actually make a huge transition into the website in terms of the Western audience and how we can make that more friendly to some of our networks. So there is going to be a 2 to 3 week delay in terms of full website maintenance, but the core items can be still available and you should have a look straight away. Besides the search engine, it's this very cool ability for you to log on and look at different signaling pathways. So example, you have a protein knockout and you come to the lab saying, oh, I saw this protein. Maybe it's kind of working out in this mechanism or this pathway. So let me look at the AMPK pathway, and you can click on the AMPK pathway, and that gives you a huge list of our mice available. And what's really cool, and I was a nerd when I saw this first, is that you get this pathway and you can just click on each of these proteins as you were just clicking anything else, and each mice that's available in this pathway or this protein comes up. So it's very user friendly and to the point. So it's kind of cool, though the interaction itself is kind of neat, and we encourage you to use it. So besides of how we're doing it and how user-friendly our website is in terms of engaging with us, we're also very personalized, and I believe we have a friendly team that you can always email or contact. And what we try to do is, even before you purchase anything, we try to help you through the process, guide you, give you strategy design, give you other tools such as sequences, primer detail, production processes, donor sequences, you name it, we never say no. If you want a different site of validation, we can give you that as well. So we're actually giving you a full benefit before purchase to make sure you're happy, and then if you're happy, we're happy, and we can provide you great service. So that is what the KOAP project's all about and what we're achieving. But I want the last 3 slides to look at other things that GemPharmatech can provide you. And we're very excited about our Cree strains as well. They're very novel. I know a lot of Cree strains are widely used in different places, but our Cree strains are very novel in terms of what kind of protein drives them, and genes drive them as well. So we can look at different fields. So for example, immune systems, we've got the classical Crees, but we also have 40 plus Crees that are not used, never been used, or even are very novel. So please talk to us. We can even look at bone, muscle, and we can generate any Cree you want. In fact, we have so many Crees on shelf now, we're actually asking academic labs to validate for us. So that's how exciting our growth in the Cree strains are. So to date, I mean, the last figure we saw in October was 192 Cree strains, and that's rapidly growing. So we can give you flox mice and we can give you Cree strains as well. And then what's exciting, and that's we also believe another platform that is world beating is our ability to use humanized immune checkpoints and to use our humanized banks in terms of generating the single target, double target, even triple target mice. This is competitively outstanding work that our scientists do. And what we have now done is also made it available in dual genetic background, so you can get it in the C57 background and also the BALB/C background. So to use this for immuno-oncology work is fantastic and is one of our flagship platforms we're very proud of. And lastly, the slide I want to talk about is that we've got more and more and more. So we also do germ-free mice, flora replantation, and different kind of germ-free experiments. You can come to us. We do disease models, immuno-oncology models, autoimmune diseases, cardiovascular diseases, osteoporosis. You can turn up and one customer asked me, hey, Pradeep, I want a tooth model. And we got an experimental team working for him. Another customer said, do you do an OA, osteoarthritis model? We said, yes, we can do it for you. So really, GemPharmatech, besides our KOAP project, is a one-stop solution for all your mouse preclinical needs. So that comes back to my kind of first few slides where I showed you, hopefully I showed you the capacity and the ability for GemPharmatech to help you as a scientist and academic. We really want you to be the kid in that sweet shop. We want you to dream big. We want you to have that academic freedom to think in a different variety and say, I want to target this, I want to do this, I'm going to GemPharmatech. And we were like, yes, we can. Our motto and slogan is models that defy the impossible. And this is no understatement. We rarely say no. And we've got a lot of tools, expertise, and backup to help you. So please be the kid in the sweet shop and come to us. And with that, I want to thank you for your attention. And I'm going to leave this slide up just for your sakes to contact us wherever you are in the world. And we wish you the very best. And hopefully we can come work together. Thank you.
Start again. Thank you very much to Mark and Pradeep for the presentation. Short and sweet. So just to follow up on Pradeep's last slide. Pradeep, do you mind sharing your last slide on the screen? Yeah, so, contact us if you have needs or inquiries. These are the information you can use, and also follow us on LinkedIn, on Facebook and Twitter. At the same time, also, as Pradeep mentioned, our website is under the current cycle of development and maintenance. So there may be a lag that you will experience if you go on our website. At the same time, though, do use the phone call and emails to contact us if you have inquiries. And then now I'm going to turn over to the Q&A session for the day. We have a few minutes to do that before we end today's event. So I'm watching in the chat, and I believe Mark and Pradeep are looking into the Q&A session. I'm going to start with a couple of questions I have received. And I think the first one is more for Pradeep. So how long does it take to deliver your knockout mouse to us?
Yeah, that's a great question. No matter where you're in the world, we deliver, and we try to deliver very rapid pace. So if we have it on the shelf, it's a matter of weeks for you to get it in terms of having the mice, or if you want a sperm or the mice, we can do both. And if it's there, it's a matter of weeks. Obviously, if we have to rederive and we have to make the mice and send it, it's approximately 3 months or just over. But if it's a brand new mouse and you want to make it, and which is very rare because our strains library is quite varied, but if it is a brand new mouse or a complex model, it can take up to 3 to 6 months or a bit over. And we have no problems in terms of global shipping. We have in the last 5 to 8 years shipped all over the world, and we can reach you whichever nook or cranny you are, we'll get the mice to you.
All right, thank you. And there's a second question. I think this is more for Mark, but Pradeep, feel free to jump in too. So the question is, you have introduced your immune checkpoint mouse models. What's your advantage?
Well, I think that the biggest advantage that we have is we've had tremendous validation through a customer base. Many people have ordered the mice and used them, so they have been validated in the hands of other scientists. And also the breadth that we have. We have over 120 different models. We have the double and triple combination. So we really are getting a more complex set. So we believe that the entire set that we have makes us unique.
Can you, so a follow-up on this question, the customer also asked, can you follow up on, downstream services that you might be able to provide or you are already providing?
Yes, actually, if you go on to the current website, there's an extensive addition of services that are available to customers working within China, but also customers from around the world can select from a large variety of experimental models that can be done. Part of our role in the US, and we should be having some press release and announcement soon, is that we are expanding animal services within the US. So soon we will have a distribution available through the US channels, as well as growing and housing mice within the US. And those will be available for experimentations, adding antibodies or small molecule screening. Those services will also be available.
Great, fantastic. I got a third question. I think probably both of you can chime in. What the other product pipelines are available at GemPharmatech?
I'll let Pradeep answer that since I've been talking so much.
Yeah, so really there's 5 different keys. I've touched upon them all. So we're looking at the knockout project, KOAP, the germ-free mice, disease model, the preclinical efficacy studies, the pharmacokinetics, and so that's something we haven't mentioned that's available as well. And then the humanized mice. We also have an exciting thing where we have a tumor bank where we look at different cancers as well that can be used. And our germ-free mice also have flora transplantation from different patients as well, which is quite cool and exciting. And then obviously, as I mentioned, we have the disease models, which is very specific. And to add on to Mark's point, most of our models, if not all, have traditional validations of qPCR, and we can also do sequencing if you want of certain things. And also we have FACS, immunohistochemistry, these validations and brochures we can make customers available to customers. And also we're very extensively networked. Our facilities are huge. So if you have a scientific technique, we can do those for validation or downstream. But again, we're well established in China and also in the world. So any technique is not out of our reach. So a client came to me and said, we want full genome sequencing of this and this and that. We were able to get it done. So downstream techniques and processes are not a problem for us.
All right, that sounds great. I'm seeing another question. Very likely this is for Pradeep, but Mark, please also feel free to chime in. So the question is, can you offer the service of vector design, using CRISPR and Cas9?
Yes.
How would you do that?
Well, how we do that? We're actually very rapid. So we talk to you at first, contact us, of course, and then we'll have a look of what you need. And what we do is we assign you with essentially a technician or a group of scientists where we can advise you, look at what you need, and then really personalize and customize the quote according to your needs and go back and forth. So we won't jump here. We won't give you a set rule. It's very personalized, and then we can talk about the services we provide to you.
Great. Thank you, Pradeep. So I'm looking into the chat box and the Q&A session again, just to make sure I didn't miss any of the questions. I'm not seeing more questions at this moment. So if we don't have more questions from the audience, I'm going to thank Mark and Pradeep again, and also thank all our participants for the day. Thank you for joining us and hearing from us. We we will look forward to hearing from you in the future. And also, as I said, this is our first one of the year for the series. So do follow us and keep up with us. We'll have more information to you in the near future. Thank you, everybody. Have a great day.
Thank you.
Thank you, everyone.